Hello, children. Welcome once again to the next module of the lesson, the fundamental unit of life. But I guess in the last module, there was some problem in the last two slides as well as in the assignment part. Yeah, where the last two slides, it was not audible as well as the questions were not visible. So in this present module, I'm just going to include both the topics as well as I'll be giving you the questions which was given in the last module. Before we begin with our new topic today, let's revise what we learned in the previous module. We had learned about the cell wall, cytoplasm, as well as few cell organelles, that was endoplasmic reticulum, Golgi apparatus, and lysosome. But we are learning once again the Golgi apparatus and lysosome because of the problem in its audio part. So in today's module, we are going to learn about the Golgi apparatus, lysosomes, plastids, vacuoles, and mitochondria, as well as the last topic of this lesson will be cell division, that is about mitosis and meiosis. Children, let's study the next cell organelle, that is Golgi apparatus, which is also called as Golgi body. Now, Golgi apparatus was first described by a scientist named Camillo Golgi, and hence this organelle got its name as Golgi apparatus. So, he was the first one to observe this cell organelle in a cell, okay, under electron microscope. Now, what is a Golgi apparatus? Golgi apparatus consists of a system of membrane-bound vesicles, cisternae, and vacuoles. Now, as you can see here in the picture, this is the Golgi apparatus, which has, these flattened sac-like structures. So, these flattened sac-like structures are called as cisternae, okay, which are in the form of poles. Then it has vacuoles, which are storage, sac like structure. And vesicles are what? These are more or less like vacuoles only, but if you take a circular like elastic thing, okay, and like made up of rubber, and if you stretch a bit, what will happen? It ends up going to be stretched on both the sides. So that shape is called as oblong. So these vesicles are what? These are oblong bags which has some material stored in it. Now, as in this picture, you can see the cisternae are arranged parallel to each other, yeah, and they're forming a bundle one above the other. Yes, so this bundle is called as a stack. What is it called? Stack. And this stack is being given another term called as cistern. Okay, so don't be confused with cisternae and cistern. Cisternae is one fold. This folded structure is called as cisternae. So there are many cisternae here, and this stack-like structure, okay, is called as cistern. Now, these Golgi apparatus, where are they present? They are present near the nucleus, okay, in case of animal cells. But do you know in plant cells, it is distributed freely as small units and they are termed as dictyosomes. So in plant cells, what is the name of Golgi apparatus? It is termed as dictyosomes. Now, the Golgi apparatus has two phases. One, can you see here? This is the cis phase and here is the trans phase. Now, cis phase is the phase of the Golgi apparatus which faces the nucleus, and trans phase is towards the plasma membrane. Now coming to the important functions of the Golgi apparatus. The main function of the Golgi apparatus is storing, modifying, as well as packaging. For example, if we take a simple example, assume that if here is the nucleus. So if from the nucleus the proteins are being synthesized, so it will come out through the nuclear pore, and through the cis phase it is going to enter, but those proteins are always in the form of vacuoles. They are surrounded, they are present inside a bag like structure. So these are going to enter through the cis phase of the Golgi apparatus. Then inside the Golgi apparatus, that is inside these cisternae, what is going to happen? A few changes are going to take place in that, and again it is going to be packaged, and again from its trans phase it is going to come out again in the form of vacuoles. And what will happen? It will go to the target site, whether it is intracellular, means if it is required inside the cell, or in any other part of the body. Now, the second one is that it also helps in, for making the complex sugars. Okay, with the help of simple sugars, and Golgi apparatus is also involved in the formation of lysosomes, which is another cell organelle which is present in the cell, which we are going to study in the next slide. Children, in the first slide, I told you that we'll be learning about lysosomes. So as you can see in the picture, here this green color structure represents what? This is the lysosome. Yeah. So lysosomes are what? It is also a membrane bound organelle which are spherical sac like structures, and they're filled with kind of digestive enzymes, and these enzymes are produced by whom? By the rough endoplasmic reticulum. Now, as the name implies, digestive enzyme means its role or its function will be what? To digest something. Yeah. So what happens? It digests the worn out cell organelles, bacteria, etc. Now, if like for example, if any foreign substances enters into the cell which is not a part of cell, it's a foreign material. So what happens? The lysosomes releases this digestive enzymes, and it degrades or kills the foreign material, maybe a bacteria, virus, or even worn out cell organelle. Worn out cell organelle means what? Sometimes the cell organelles present inside the cell gets damaged, or it becomes old, or sometimes it might also become non-functional. So in that case, the lysosomes digest those worn out cell organelles. Even if any complex substance enters inside the cell, so these digestive enzymes breaks down into simpler substances. So thus it acts like a waste disposal system of the cell. Okay, because whatever unwanted things enters the cell, it digests it. Now when the cell gets damaged, lysosome may burst and the enzymes digest their own cell. Hence lysosomes are called as the suicidal bags of a cell. What does it mean? It means that when the cell anyhow gets damaged, then what happens? The lysosome also bursts, and what happens as a result? The digestive enzyme which is present inside this lysosome will be expelled out, and it digests the lysosome itself. So hence lysosomes are called as the suicidal bags of a cell. I think till here, each and every cell organelle is clear to you all. Do keep on revising the topics which we have covered till here. In the next module, we'll be learning few other cell organelles, and at the end of the session, we'll be writing the question answers, as well as few question answers will be given to you, which you need to write by yourself. Thank you. Children, now we are going to study about the plastids. Okay, now this is also a cell organelle which is found only in the plant cells. Yeah, and it is a double membrane bound organelle found in the cytoplasm. So, plastids present in animals? No, they are not present in animals, because plastids are it's the organelle which provides color to the plants. Now, apart from providing color, plants make and store food in plastids. Okay. Now, plastids have their own DNA, that is the deoxyribonucleic acid as well as the ribosomes. Now, plastids can be classified into two types depending upon its function. First one is the chloroplast and second is the leucoplast. Yeah. So, chloroplast is what? It is a type of chromoplast. Don't be confused with the term chromoplast. Chromoplasts are nothing. It's a type of a plastic which are colored. So the colored plastids are called as chromoplast, and one type of chromoplast is the chloroplast. Yeah. Now chloroplast has a green color pigment. Yeah. The chloroplasts are the plastids containing the green pigment called as the chlorophyll, which is very essential to carry out the process of photosynthesis, because if chlorophyll is not there, the process of photosynthesis won't happen, won't take place. Why? Because chlorophyll absorbs the solar energy. Yeah. Now chloroplasts also contain orange and yellow pigments, but on the other hand, leucoplasts are what? They are white and colorless plastids. So as they are colorless, will they provide color to the plants? No. So their role is not to provide color but a different function, that is to store. What to store? Starch, oils as well as protein granules. Now plastids have many membranous layers which are embedded with the surface called stroma, as you can see here. Now this is the picture, okay, of the plastids. Now here it's a double layered membrane that is having the outer membrane and the inner membrane here, and in between that there is one, the slight green color, that is the intermembrane space, that is the space between these two membrane, that's outer and inner membrane. Then we have this light yellowish in color, this is the liquidy what we say, aqueous fluid, yeah, uh, filled inside this cavity called as the stroma. And inside this, can you see this small disc shaped structure? So this one single disc shaped structure is called as the thylakoid. What is it called? Thylakoid. And these thylakoid are forming stacks, that is they are present one above the other, forming a stack or a bundle. So this stack of thylakoid or a bundle of thylakoid is called as granum. And can you see there are many granum present? So all these granum are called as grana, and they are interconnected with each other with the help of lamella. I think this is quite clear to each and everyone till here. Children, now we are going to study about the vacuoles. Now, do you know when the plant cell was observed under electron microscope, scientists first thought that there is an enlarged empty space. But cell being an important part of organism, how an empty space can be there? Yes. So, well, there is no empty space in the plant cells. Actually, it is a fluid-filled cell organelle. And what is this called? This is called as the vacuole. Now, in this image, you can see animal cell as well as the plant cells both possesses this vacuole, but there is a difference, that is in animal cells the vacuoles are very small and there are several, but in case of plant cells there is one large vacuole. Okay, as well as what happens now this vacuole, it is used for storage, so that's why it's a storage sac, yeah, that contains nutrients, water as well as waste, as well as in case of plant cells, what happen, it is used to store water and push against the cell wall and keeps the plant rigid. That I'll explain you later on. Now the term vacuole has comes from a Latin word vacus, okay, that means empty. Now, vacuole is filled with a fluid, and that fluid is called as what? Cell sap. What is it called? Cell sap. Now, do you know in some cells the vacuole occupies 50 to 90% of the cell volume. But why is the vacuole so big in plant cells? Now, unlike animal cells, yeah, plant cells don't have an excretory system. So in that case, they need to store the unwanted, waste, unwanted products somewhere. So that's why when wastes are produced, they are stored temporarily or permanently in the body, and that is where in the vacuole. So vacuoles store the waste products, whereas in some cases, large waste molecules are also broken down here. Now, and vacuoles not only store waste materials, but it also stores some useful materials. For example, amino acids, sugars, organic acids, some proteins which are essential for the functioning of the cell. Okay, and one more very important role played by the vacuole is to provide what turgidity and rigidity to the cell, firmness to the cell. Yeah. So, what is this turgor? What is turgor? Turgor is what? When a plant absorbs water through the roots here. So in that case, what happens? The volume of the vacuole increases. So if the volume increases, it is going to occupy more space and it is going to push the cytoplasm against the wall. Yeah. So as a result, a pressure will be created on the wall, and that pressure is called as turgor. Yeah. Now, just assume this. Now, when there is a water absorption, so what will happen? The vacuole volume is going to increase. So, as a result, the pressure is going to increase. That is, the turgor is going to increase. So, when turgor reaches the maximum level, yeah, that is, this pressure is maximum. After that, if the pressure has been applied, the cell is going to burst. So when this turgor reaches the maximum, the water uptake, stops by itself. And on the reverse case, what happens when the plant loses water by the process of transpiration? So what will happen? The vacuole volume decreases, so the turgor is also going to decrease, and in that case, what will happen? The water uptake will begin. So I think till here it's clear. Now, the vacuoles, it has also been observed in a single-celled organism such as amoeba. So we have already learned that amoeba has a food vacuole for what? For ingesting as well as digesting food. Yeah, and it also has one specialized vacuole, that expels the extra water as well as the waste from the cell by continuously contracting. So as a result, it contracts continuously. That vacuole is termed as contractile vacuole. So amoeba has two vacuoles. One is the food vacuole and the second one is the contractile vacuole. Now the last cell organelle is the mitochondria. Okay, so till now, whichever cell organelle we have learned out of all, mitochondria is the most important cell organelle. Without mitochondria, the other cell organelles or the cell cannot function. For example, if there we have a lot of electronic gadgets, yeah, but if there is no power, no energy, yeah, means in the form of electricity is being provided, will the gadgets work? No. So same here, if mitochondria was absent, the cell organelles or the cell won't function. Yeah. Now, so that's why mitochondria is called as the powerhouse of the cell. Now, first of all, mitochondria was first discovered by the scientist Colliker in the year 1880 in the flight muscles of insects. Now, flight muscles are what? They're used to control the wind during flight, and the term mitochondria was given by Benda in the year 1887. Now, the shape of the mitochondria can vary. They can be round tube-like, yeah, or they can be cylindrical, yeah, or rod, like tube-like means rod-shaped, yeah, that provides energy to the cell in the form of ATP. And what is the full form of ATP? Is adenosine triphosphate. And why this ATP is required? Which it is required for performing different chemical activities for the survival of life. sustaining of life. So thus mitochondria is called as the powerhouse of the cell, and ATP is called as the energy currency of the cell. Now, when mitochondria was observed under electron microscope, it was found to be a double membrane organelle. Okay, now in which the outer membrane, okay, as you can see here in this image, it has two membranes, outer membrane and inner membrane. So outer membrane was found to be porous, through which the transport of nutrients could take place, as well as the inner membrane had deep folds. Yeah, and these folds were called as the cristae to increase the surface area of the mitochondria, so that a large number of ATP can be generated. Yeah, now this ATP is being produced by mitochondria, which is required for the cellular respiration. Yeah, now mitochondria have their own DNA and ribosomes, hence they are capable of producing their own proteins. They are not dependent on any cell organelle. Okay, but the DNA which is present in mitochondria is different from the DNA which is present in the nucleus. And do you know, it is believed that mitochondria have originated from the primitive bacteria. It was found, means, outside the prokaryotic and eukaryotic cells, and it was later on engulfed by these cells. So according to this, what happens, it is being thought that the bacteria which was engulfed by these cells remain inside the prokaryotic or the eukaryotic cell at same point. So that's why it's thought that mitochondria have their own DNA and ribosomes. Now, I think till here, each and every cell organelle with their structure and functions are clear. If you have any doubts, please mention or note down in your notebook. Once we meet, we are going to discuss about that. Now, in the next slide, we are going to learn about the cell division. Now, the last topic of this lesson is the cell division. Now, children, we all are aware that all multicellular organisms, okay, starts its journey from a single cell. Yes. So, how does a single cell goes on to form such a large organism? So, that is nothing by but by the process of what? Cell division. So, what is cell division? We are going to study about in this present slide. Now, cell division happens when a parent cell, yeah, divides into two or more cells, and those cells are called as what? Daughter cells. Now, all cells reproduce by splitting into two, where each parental cell, that's the parent cell, gives rise to two daughter cells. But not in all, that is, this happens in case of mitosis, but in meiosis, instead of two, four daughter cells are formed, which we'll be learning in the next slide. So how can we define cell division? Cell division is the process by which a cell called what? The parent cell, yeah, divides into two cells called as the daughter cells. Yes. Now, during cell division, all the tiny elements of the cell, whatever components are present inside the cell, they also divide, including the cell's chromosome, nucleus, mitochondria, and many more. Yes. Now, cell division, uh, uh, in cell division, these newly formed daughter cells could themselves divide. They further divide and grow, giving rise to a new cell population that is formed by the division and growth of a single parental cell and its descendant. Descendants means the offspring, that is from a single parent. As you can see here, okay, this was a single cell where it is being divided into two separate cells called as the daughter cell. So this keeps on going. Okay, so what happens, such cycles of growth and division allows a single cell to form a structure consisting of millions of cells. And do you know, in every 24 hours trillions of cells divides in our body. Types of division. There are two types of cell division. Yeah, the first one is the mitosis, and the second one is meiosis. So what is mitosis? Mitosis is a process where a single cell divides into two identical daughter cells, yeah, where the newly formed DNA is separated and the two new cells are formed with the same number and kind of chromosome as the parent nucleus. Now this is the process of mitosis being shown diagrammatically. Here is a single cell. So in the second phase, the DNA replication takes place here. And after that, what happens? The nucleus divides and then there is a division in the cytoplasm. And as a result, two daughter cells are being formed. And here the number of chromosome in parent cell as well as the two daughter cells formed will be same. Now in human cell, there are 23 pairs of chromosome. Now in case of mitosis, what we can see that it is a process where a single cell, this was a single cell, it divides into two identical daughter cells. And what is the main purpose of mitosis? Its main purpose is for growth and to replace the worn out cells, that is the damaged cells. Yeah. Now this what type of cells show such type of division? Cells like skin cells, yeah, blood cells, bone cells, organ cells, as well as the plant cells shows such type of division. That is a process of mitosis. Now mitosis, here we are learning only the definition, but there are different stages of mitosis and meiosis. So I'll only tell you the names of those stages. So mitosis have has five different stages. The first one is interphase, then prophase, metaphase, anaphase, and telophase. Okay, so what we can say that mitosis is a way of making more cells, because from one single cell two daughter cells was formed, and we learned that it is a process for growth, for increasing the number of cells. So it is a way of making new cells, more cells that are genetically same as their parent cell. So what happens? First the nucleus of the cell is dividing, and then the cytoplasm divides, yeah, that is called as cytokinesis. Yeah. And what happens? How many daughter cells are formed? Two identical daughter cells are formed. And mitosis is also known as somatic cell division. What is the other name of mitosis? It is also called as somatic cell division. Now coming to the next type of cell division, that is the meiosis, which is shown here diagrammatically. Now meiosis is a type of cell division in sexually reproducing organism that reduces the number of chromosomes in reproductive cells from diploid to haploid, leading to the production of gametes in animals and spores in plants. So it results in the formation of how many daughter cells? Four daughter cells, and from one single parent. From one single parent, how many daughter cells are being formed? Four, and they have haploid number of chromosome. That is, each daughter cell will have half number of chromosome compared to the number of chromosome present in the parent cell. So meiosis is what? Meiosis is a specialized form of cell division in which a single cell, yeah, divides twice. There are two stage division happens in two stages to form four daughter cells, yeah, that possesses exactly the half number of chromosome of the parent cell. Therefore all the four cells are what? Diploid or haploid? They are haploid, that is having half the number of chromosomes, yeah, and will also contain the half the original amount of genetic information. Now meiosis, now it occurs in reproductive cells like sperms, eggs, as well as spores in plants. Now meiosis, it consists of two successive cell divisions. There are two stages of cell division that resembles like mitosis only, but here the chromosomes are duplicated only once. Here it was replicated once, so here also it in the first stage it is being replicated, but in the second time it's not replicated. So gametes will have now what they have half the number of chromosomes. Yeah, so that's why another name of meiosis is what it is also called as the reduction division. Yeah, why it is called as the reduction division? Because the number of chromosome is reduced to half in each of the four daughter cells. Now, we'll learn about the different sub-stages of meiosis. One is meiosis 1 and second is the meiosis 2. Now, in meiosis 1 can further be broken down into four sub-stages, that is same as mitosis, that is prophase, but here it will be prophase 1, then metaphase 1, anaphase 1 and telophase 1. And in meiosis 2 will further also be divided into four substages, that is the same stage, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. So uh, in uh, in this standard, that is in ninth standard, we are going to learn till here only. We'll be learning in detail in higher standard, that is in 10th grade. Now, so what is the, before we conclude this topic, let's just recall the difference between the mitosis and meiosis. In mitosis, um, from one parent cell, how many daughter cells have been formed? Two. But in case of meiosis, from one parent cell, how many daughter cells have been formed? Four. Yeah. Now here the number of chromosomes, yeah, are same, but here the number of chromosome reduces to half. Now meiosis has how many division? One division, but in mitosis have one division, but in case of meiosis, how many divisions are there? There are two consecutive divisions. So I think children, it is quite clear to each and every one. As I told you earlier, if you have any difficulty, please write your doubts, your queries in your notebook, and we'll be discussing later on once we meet. Now, there are few questions which we missed, okay, in the last module as it was not visible. Questions are, first one is what are cell organelles? The second one is explain the structure and the function of Golgi apparatus. Third one, why lysosomes are the tidal bags of a cell. Fourth one, what is endoplasmic reticulum? Name the two types of endoplasmic reticulum. And the fifth one is which organelle is associated with protein synthesis. In few questions, children, you need to rack your brain and write in your own words. So write all the answers in your notebook. Okay. So till then, revise your topics whichever we have completed. Uh, okay, goodbye. Have a great day. Thank you.